Oh, hey there. You're looking at my family tree, aren't you? Here, come along with me. Come here. Come here, come here. You see? Come here, come on. That was me as a little boy. See how cute I was? It's all due to this. And now, we're going to talk about Mendel's gene idea.
Mendel, though, was that he wasn't the first to have questioning about genes. He just formed a big process. But one of my colleagues has some information that took place before Mendel came up with his big idea of the gene.
Hippocrates proposed that each part of the body produces something which is then somehow collected in the semen. Darwin proposed a similar idea to Hippocrates, naming it pangenesis. He also concluded that crossing has a unifying effect. He used Aristotle's argument against pangenesis for it, saying that gemmules, which were gametes, did not necessarily express themselves in the first generation. Aristotle argued against Hippocrates and Darwin's ideas, giving the example that children sometimes look like ancient ancestors rather than parents. Eventually, he came to accept the hypothesis of pangenesis, but knew that the real answer would not be a simple 1-to-1 ratio. His general conclusion was that what is inherited is not characters themselves in any sense, but only the potentiality of producing. Kohlreuter concluded that the fertilizing agent in plants was the fluid released on the stigma, rather than a formed element from a particular pollen grain. He also concluded that more than one pollen grain is needed for the production of a normal seed, which delayed the development of a clear understanding of fertilization. The latter half was also accepted by Darwin. Gartner conducted thousands of crossing experiments, but did not add much to Kohlreuter's theory other than that the F2 generation has a greater variability compared to the F1 generation. Herbert contributed that crosses between species are unsuccessful or produce sterile offspring.
Wow, now that was crazy. But I think our director kind of confused Herbert with Sherbert, so there might be a little bit of a mishap there. Now, we're going to have a very fine young man, very intelligent. He's going to be talking about Mendel's research and what he kind of discovered during his process. When Mendel conducted his experiments, he designed his experiments carefully, obtained large amounts of data, and used math to show that his results correspond with his explanatory hypothesis. Mendel chose the garden pea because they were available from sea urchins with various shapes and colors, and the peas can self-pollinate or cross-pollinate. Mendel could cross-pollinate the plants whenever he liked because he removed the anthers of one plant with the pollen not open and then transferred the pollen to the receptive stigma with the paintbrush or on anthers. Mendel also had 4 concepts with his model. His first concept was that alternative versions of genes account for variations in inherited characters. His second concept was that an organism inherits 2 copies of a gene from each parent. His third concept was that 2 alleles differ at the locus: the dominant allele, which determines organism's appearance, and the recessive allele, which has no effect on the organism's appearance. His fourth concept was the law of segregation. Mendel formed 2 laws to explain his research: the law of segregation and the law of independent assortment. Mendel's law of segregation states that the 2 alleles in a pair segregate into different gametes during their gamete formation, while the law of independent assortment states that each pair of alleles segregates or assorts independently of each other pair during gamete formation, which applies when genes for 2 characters are located on different pairs of homologous chromosomes, or when they are far enough apart on the same chromosome to behave as though they are on different chromosomes.
In 1900, botanists de Vries, Correns, and von Schermach independently rediscovered Mendel's work while doing their own work on the laws of inheritance, which allowed Mendel's work to be put into the physical world. In 1902, Walter Sutton observed segregation of chromosomes during meiosis that matched the pattern that Mendel discovered. And later that year, Archibald Garrod observes a disease that is inherited according to more Mendelian rules. In 1909, Wilhelm Johansen coins the word gene to describe the Mendelian unit of heredity, which came up with terms of genotype and phenotype. In 1944, Barbara McClintock discovered that genes can move around in chromosomes. And then in 1973, researchers fused fused a segment of DNA from frog and E. coli. Frog DNA was copied and directed the production of a specific frog protein.
Genes have also led us to discover where maybe our hair color comes from, our skin color, physical traits. Those are all related to genes. And by studying those, we can determine diseases such as cancer, sickle cell disease, those inheritable diseases. And it helps us understand so we can get treatment earlier on and be more cautious of what may happen. Now, there are some genes, especially for us men, who may go bald. Like Mr. Johnson, he was unlucky. He got a gene with bald hair, but on another gene, he got a luscious beard. That's very resourceful for him. His hair went down from his head all the way down to his beard, and now he's well acclaimed for his beard. Next, there's new ideas rising that with CRISPR, we are able to alter our genes. So maybe we'll be able to pick our children one day, which would not be great because we're getting rid of reproduction. But it could help us alter diseases so we don't have those diseases anymore and we can help combat those. So there's a good side and a bad side. Good side of helping people with their diseases, but also the bad side of making human beings and possibly creating an
We can
organ.
closely monitor family history in order to catch cancer when people start showing the first symptoms or in order to do a routine screening. There have been instances when the information was newer where some things may have been missed that could have been avoided, such as losing someone to cancer because they weren't monitored closely enough.
The benefit of this knowledge helps you better understand your health and what choices to make in order to keep you healthy. Genetics can also track a disease that runs in the family, and with this, you can learn if you will get the disease. Genetics explains why you look like people in your family as well.
Ancestry companies say they can inform individuals of their ancestry and health risks, but scientists argue that results are not always accurate and can lead individuals to make questionable health decisions. These companies are also a threat to privacy as they have unclear laws about what they are allowed to do with consumers' DNA. Researchers have been creating superviruses to better understand the flu and how to stop them, but the public has concerns about what to do if the virus escapes or if it is weaponized. There's a large continuing debate about GMOs and whether or not they are safe.
Oh, you're back. Well, sorry, I was gone a little bit doing some research on some things, but that was a pretty good amount of information we just got there, right? Well, we're about to wrap it up with some historical figures that have shown our generations and possibly future generations how genes work.
In 1858, Charles Darwin and Wallace discovered how new species arise by evolution and how natural selection occurred. In 1865, Gregor Mendel is known as the father of genetics for his work with pea plants and their inheritance and genetics. In 1944, Avery, MacLeod, and McCarthy show that DNA was the factor responsible for genetic inheritance and evolution of the bacterial strains discovered by Griffith. In 1953, Watson and Crick determined the structure of DNA. In 1972, Berg constructed the first recombinant DNA molecule. In April of 2003, the Human Genome Project was finished.
The gene idea has helped shape us into who we are today. Without genes, we would be just like some dogs here. But genes give us variety from our mother and our father, and we are closer to our siblings. Without genes, we'd all be the same, and we wouldn't be different. Now that would suck. But thank goodness we aren't walking around on 4 legs like this little guy here. Thank you.